So, good morning, everyone. I hope my screen is visible properly. Okay. So, first of all, my sincere apologies that I was
I was
not
not
able to
able to
join
join
personally.
personally.
I
I
wish I
wish
would
I
have.
would have loved to talk in front of you because that would have been much better actually. And a big thanks to the organizers for giving me this opportunity to
Talk something about our research, that what we are doing and how we are working in the aspect of biotransformation of food and feed.
feed.
So,
So
before
before coming directly to the talk subject, I wanted to introduce my institute to the candidates. It might be, I would love if you would like to visit our institute. Our institute is National Agri-Food and Biomanufacturing Institute under Department of Biotechnology, Ministry of Science and Technology, Government of India. And the vision of our institute is food and nutritional security through agri-food biotechnology research and innovation. So
we
we
have
have
two
two
major
major
departments, food and nutritional biotechnology, agriculture biotechnology, and a well-supported biomanufacturing unit, which is being recently inaugurated. So we, starting from basic science to technology development to scale up, we have facilities for all of them. So now going directly to biotransformation, I just wanted to give a key introduction of functional food, what are functional food, and how biotransformation helps in production of functional food and feed, and what is the importance in the global market. So we are, functional food are food component that has a specific health role or a functional property, like food with a antihypertensive property or food with more of vitamin, specific vitamin like vitamin B12, or food that can help in improving mood, weight management, or any particular health like digestive issue or heart disease, or even nowadays we have food for skin and improving skin and properties and beauty products as well. So there are many players in this functional food industry. If you these are some of the important players globally, like PepsiCo, and you definitely see some ads that you eat particular food, you will get weight loss, or this particular food will improve the brightness, etc. So these are nothing but functional food. Now, if you see how functional food is different from nutraceutical, people generally get confused that what are functional food and what are nutraceutical, and are they both same or they are different? So there is a thin line of difference between both. Nutraceutical is a commodities derived from the food. Like if you talk about peptide or you talk about a particular polyphenol or only vitamin B12 capsule, that will come under nutraceutical. But if a food component like a curd with improved vitamin B12 or a fermented soybean product improved with bioactive, that is called a functional food. So microbial strategies or biotransformation can be useful for developing functional food with improved specific components. Now, if you see the global market, definitely the functional food market is increasing, and it is much increasing at a faster rate, specifically after COVID, when people are looking for food with improved immunity, food with improved several other aspects, and among them, plant-based fermented food, functional food are improving more. So this is a trend which is there, and it is expected to grow much in 2032 or even beyond that. So, if and the second important aspect is the regulation, like we definitely take medicines for hypertension, diabetes. Now, there are regulation which governs this application of functional food or delivery of this functional food. Like you have a Foshu in Japan, like they have developed several food products, like food with improved gastrointestinal condition, food with that related to blood cholesterol level or sugar level, blood pressure. Like there is a product in Japan called sour milk. It's called calpase. It's a fermented milk product which has two peptides, VPP and IPP. And these two peptides have been clinically proven, not these two peptides, the fermented milk product enriched with these peptides have been clinically proven to reduce hypertension. So now, if a food is giving you some health benefit, definitely it has to be governed with a regulatory agencies. And like FOSU in Japan or FSI in India are working towards creating regulations for new foods which are targeted for improving a particular health conditions. Now, if you see fermentation technology, or why fermentation technology? Because fermented food or such things are done by microorganisms like lactic acid bacteria, yeast, or bacillus. And these organisms, they produce different enzymes or metabolites which helps in transformation of food to a functional food. By, like, for example, I'll give you some examples in next few slides where protein or carbohydrate can be hydrolyzed or bioconverted into nutraceutical or functional food based products. Now, in India, especially, if you see, and not only in India, in other countries as well, fermentation technology is a traditional biomanufacturing. Actually, we call it a traditional biomanufacturing because curd you're hearing curd or fermented milk products since many years, even in Mahabharata or Ramayana, and is other product like wine you're hearing since many years. So this is nothing but a traditional biotechnology which is there, and we are trying to improve it by understanding what are the role of microbes, how this microbes can help in fermentation, but for development of a standard product, but also a product with improved health benefits. How we can design this product with improving a particular metabolite which can target a specific health condition. Now, if you take our country, we are blessed actually. If you see, coming from north to south or east to west, every district they have a unique food system, and every district have a unique traditional food system. Like food is prepared traditionally, and if
Amit
you
sir,
focus, yeah,
sir, slides are not moving.
okay,
Yes, and sir, do it on full
slides are moving
screen.
now. Which you can see which slide? I am in the seventh or eighth slide.
I think you are on third, fourth.
This is the
And
first.
full screen, sir.
Yeah. Now it is full screen.
Not yet, sir.
Then I have to change it 1 minute.
No issues.
Oh, you would have called me. Now it is full screen. Is it full
Not
screen
showing,
now?
sir. Not showing, sir.
You cannot see the slides also.
No, sir. No, sir.
Oh.
I think, sir, you can start again. You just stop your screen
Yeah.
and restart. Now we
what
can
you're
see your slide, but no, it is not on full. Yes, it is fine, sir. Now, please.
Moving? It is moving?
Yes, moving, sir. Yes. Thank you.
Okay, great. Finally. So, I have already told over these few slides. Let me go directly to this picture. So, and now you can see our Indian map and different type of traditional food. That slide. Hello. Am I audible?
Yes, sir. Perfectly.
You can see the slide which shows traditional fermented food pictures.
Yes, we can see
Okay, great.
slide fermented foods of India. traditional.
Okay. So, if you take our country, we are blessed to have diversity of fermented food. Actually, if you go from northern part of India to south or east to west, the diversity of fermented food it depends on the traditional knowledge which is there in every district. If you go from one district to another, even if one particular fermented food is produced in a different way, and even if you go to the northeastern part of India, uh, the diversity of these fermented foods are much higher. Like you have this fermented hard cheese product, or you have fermented fish product, fermented bamboo shoot, or even fermented soybean product, which is alkaline fermented product where bacillus or some other organisms are there. Now there is other fermented fish product. If you will be surprised to know that these products are kept in underground. It is a fish. Fishes are generally crushed. They are tight air packed in clay pot actually, and they are kept in underground condition for a period of 1 year. And then specific biotransformation or microbial population, there is a succession in the microbial population which impacts the metabolite of fish protein or fish lipids or anything. So what is important here, why you want to say here is, like, if there is a huge diversity of fermented food in the country from in different region, definitely the microbes associated with these traditional food will be different. And the diversity of microbes, it impacts the transformation which takes place during fermentation. Like if you take two different strains of a particular organism, like Bacillus subtilis 1, Bacillus subtilis 2, they may have a similar taste in the product, but they will definitely have different impact on production of specific metabolites. So there is a great opportunity if of isolating novel strains, applying these novel strains from huge diversity of fermented food, either in terms of either from any region or any other region, for applying them for development of functional food. Now, what is the transformation which can take place actually during food fermentation? If you take this is an example of a legume. Uh, Similarly, it can be in other product as well. Like if there is a protein-rich fermented product is there, like soybean, fermented milk, or fermented fish. So definitely, if the microorganisms associated with these food are producing proteases, there will be production of several peptides and amino acids. And then depending on the sequence of these peptides, they may have different type of health benefits. Similarly, there is possibility of hydrolysis of carbohydrates or lipids, or the most importantly, bound polyphenols to free form. Now, it again helps in improving viability of particular metabolite, or there is possibility of production of biogenic amine. So these are the first 5 or 6 is biotransformation of the food substrate like protein, carbohydrate, lipid, polyphenol. So, but there are also possibility of production of metabolites directly from the organism. Like earlier was like the microorganism will produce enzymes. Those enzymes will act on the food substrate and transform them into some other product. But the organism may also produce metabolites like bacterocins, GABA, surfactants, vitamins, fibrinolytic enzymes, which can again improve the functionality of a food. Like if fibrinolytic enzyme, if a organism produces fibrinolytic enzyme and it is dominant in a traditional fermented food product, those products can be useful for heart associated problems. The third and the more important problem with this biotransformation during food fermentation is reduction of anti-nutrient factor. Like if you take an example of a soybean is rich in many anti-nutrient factors like there are lectin, and there are trypsin inhibitors, and some other protein which are having anti-nutrient property. So by the hydrolysis of enzymes, hydrolysis of the enzymes that are produced by microbes, these anti-nutritional factors can be broken down, and you can get a product which is having reduced or no amount of anti-nutritional factors. So how actually, when we say how during food fermentation or biotransformation, you can get product with improved health benefit, this in global scenario, people have worked on these aspects. Like in India, definitely we have worked, but in global they have even reached to clinical level. So like people have worked on production of fermented food rich in CLA, and that has helped in by animal model it has been shown that it can reduce anti-inflammatory property, or it can have beneficial effect during cardiovascular diseases. Similarly, polyphenols when I say it is nothing but fermentation will improve the viability of polyphenol. So if the viability of a polyphenol is increased, the functional property associated with that polyphenol will increase. So a food which will have more of antioxidant, more of antimicrobial or other properties after fermentation. Now, similarly, if it's a protein-rich food, definitely the food will have more of peptides, and then these peptides, depending on the sequence of amino acid within them, they can have different property like antioxidant and AC inhibitory, like antihypertensive, anti-diabetic, etc. So similarly, microbes can be designed, or microbes consortia can be designed that can help in fermentation, not only to give the taste flavor which you get from traditional food, but you will also get functionality which you sometimes you get or sometimes you don't get during traditional food fermentation. Now, the key one main organism group of organism which does this job is the group of lactic acid bacteria. Again, there are many lactic acid bacteria. You need to select. Like there are some which are not appropriate also for food fermentation, like a group of Enterococcus, which also comes under lactic acid bacteria, but they have been associated with vancomycin resistance. Or there are some organisms as well, but there are a group of organisms like lactobacillus, pediococcus. People have isolated them and from traditional fermented food and applied for production of fermented food or even functional food with many properties, like definitely flavor, taste, texture. That is one part. But if you want to go for functional food with specific health benefits, so these organisms have been explored in different industry, dairy industry, fish industry, bakery, wine industry, and many other products. So these are like specific examples. These are some specific enzymes which this lactic acid bacteria produce, like levain sucrase or linoleic isomerase, glutamic decarboxylase, which helps in production of these metabolites like EPS, CLA, or GABA, or even bacteriogens, which helps in improving the functional property. So these are some set of enzymes which I am showing. There are many other enzymes that are produced by lactic acid bacteria and other group of organisms, which improves specific. Like for example, if you want to produce a fermented food or functional food, which produces which has to be rich in GABA. So you select a organism which produces this particular enzyme, which helps in production of GABA. So like this starter culture selection or a fermentic consortia of production will depend on your need or your objective. So these are apart from lactic acid bacteria, yeast are very important in functional food industry, where yeast has been explored for probiotic, folate, or many other compound, or even for production of different enzymes for transforming the food substrate into bioactive form. So actually, in our lab, we focus more on bioactive peptides. So I will be discussing around 20-30% of how bioactive peptides are important and how starter culture can be designed for production of bioactive peptide for specific property. Like if you take examples of different type of protein-rich fermented food, like soybean, fish, fermented milk, or fermented meat product. So specific strain, like if you take fermented soybean can be fermented using lactic acid bacteria for acidic fermentation. Fermented soybean can also be fermented using Bacillus subtilis for alkaline fermentation. So definitely the organism will give you different product from the same substrate, but the protease machinery, the specificity of proteases that are produced by specific strain of that particular bacteria will result in production of different type of bacteropeptide. And it is not one peptide or two peptide. Like if there is a peptidome which has 200 or 300 peptides, the produced on fermentation. So the peptide, the most peptide, if they have one particular property, the food will be trending towards that particular property. Like for example, in a simple way, like if 200 peptides are produced during soybean fermentation, And out of 200 peptide, if 10 peptides have AC inhibitory property and one or two peptides have antioxidant property, so that particular organism responsible for enriching AC inhibitory peptide during food fermentation, and that will bring a product which is having antihypertensive property. So it depends. You have to screen a lot of organisms for finding a specific protease producing organism which does a particular job which you need. Either you want to improve anti-diabetic peptide or anti-cancer peptide or antihypertensive peptide, that has to be dependent on the specific strain. Now, what we did actually when we understood, okay, in our preliminary study, we found that, okay, two different Bacillus subtilis, Bacillus subtilis 1 and Bacillus subtilis 2 does different job. Like they, even though they give 50% soybean hydrolysis, but the type of peptide produced using two different strain is different. And even though the food is having similar taste, similar flavor, but the type of peptide decides the functionality. So this is nothing but a fermented soybean product of Sikkim. It is called kinema, which is an alkaline fermented product, which is produced. Major producing organism is Bacillus subtilis. So when we understood, okay, different strains are responsible. So different strains might be enriched in different season because they are all traditional fermented food. Different season may have different impact on microbial population at strain level, and that may have different impact on production of metabolites. So we collected samples from different season and isolated organisms from different season, identified them, screened many of them, and then applied them for fermentation. So our main objective, see, for example, this particular fermented food production, it needed a amylase producing Bacillus subtilis, which ferments and produces the mucilage, which you can see in the picture. But apart from that, our objective was to produce a product where this fermentation takes place, and also there is increase in free polyphenols and bioactive peptide production. So that's why we selected these three enzymes which are important for soybean fermentation: protease, beta glucosidase, and amylase. And based on that, we screened these organisms and we selected two of them which does the job. Now, our second objective was also to understand that when we collect samples from different seasons, how the microbial population changes. Like these are traditional fermented soybean product picture you're seeing, and then in the left side there is a metagenome data which shows the diversity of organism in different season actually. So when you see different season will have different diversity at species level, you can see here. And more important part is, like, when generally we don't consider many hygienic conditions when we have produced it at traditional level, so you find many pathogens as well. We found Bacillus cereus, which is dominant in December and January. We found other organisms which are dominant in one particular season. Like it depends on many factors, the presence of these organisms. And many of these organisms, like protease, Enterococcus faecium, Staph aureus, are included in critical and high priority list of antibiotic resistant bacteria by WHO. So definitely, we are rich in traditional food, we are rich in many things, but our main objective is to avoid these organisms, so the common people who are using it or consuming it should not enrich these pathogens in the product. So our first objective is to develop a product which has hygienic, and then add protease or beta glucosidase producing organism which improves the functionality of the food. So after a strong screening procedure and fermentative property, improvement of antioxidant and AC inhibitory property, we selected these two strains, Bacillus subtilis KN2B and Bacillus subtilis KN2M. And these two organisms, we did the whole genome analysis, and we found how it is similar to similar product which is consumed in Japan. There's a fermented soybean product which you will have heard, natto, and there are other fermented soybean product which is consumed in Korea. So we actually collected genome of Bacillus subtilis which are responsible for similar product from different countries, and did the comparative analysis. We found that our Bacillus subtilis K2B was found to be similar to Bacillus subtilis natto, West 195 strain, which is popularly used in Japan for production of natto. And this second strain K2M was similar to a Bacillus subtilis strain, which is applied for fermented soybean production in a fermented soybean product consumed in South Korea actually. So what we did, we wanted to see how these strains are important and responsible for biotransformation. So when we did control fermentation using two strains of Bacillus subtilis, you can see the Venn diagram. There is BS2B and BS2M. So this is nothing but Bacillus subtilis 2B, Bacillus subtilis 2M. And then we, in the metagenome data, we found Bacillus amyloliquefaciens and Bacillus licheniformis are also dominant. So we also selected one strain from each to see how they affect the peptidome and other metabolites during biotransformation. And after fermentation, the taste was almost similar in all four. But if you see, when we did the peptidome analysis, we found that many bioactive peptides are unique in each product. Like if you see the numbers in the Venn diagram, which are single actually, 104, 1240, 203, 153. So these are nothing but unique peptides in that particular product. So if you take Bacillus licheniformis, a different species altogether, you have more than 1000 unique peptides in the product. If you take Bacillus subtilis strain only, you have more than 150 or 200 unique peptides in the product. So that actually, even though the similar taste, the presence of unique peptide, and if those unique peptide are having a functional property, any property, the functionality of that particular product will get inclined towards that particular health benefit. So what we did, we screened for antioxidant peptides or peptides which are able to reduce the activity of myeloperoxidase, an enzyme which is associated with oxidative stress. We found these two peptides at the end. Actually, this you can see the sequence like Bacillus subtilis fermented product. This one particular peptide which was found to be able to be having antioxidant property and ability to inhibit the activity of myeloperoxidase. So similarly, these peptides are from different. We also shown that which precursor protein from where they are coming, like beta-glycinin, glycinin, lipoxygenesis, lectin. So that you can see one more thing, that once soybean is hydrolyzed, lectin, a antisense factor, is also hydrolyzed, and so the antisense factor associated with lectin is reduced. So fermentation not only produces this peptide, but these peptides are produced from antisense protein which is present in soybean. The second figure which you're seeing the metabolite profile, actually, the metabolomics data, where we have shown specific metabolites which are produced by fermenting soybean using 4 different strains, the 4 subclase or licheniformis. So even the presence or dominance of specific metabolites, it varies when you use different strains. Like when you use different strains of Bacillus subclase, licheniformis or amyloliquid. So that is why it is very, very important at strain level, not only genus and species level, at strain level to select a culture which does the desired transformation at higher efficiency. So this, if you take an example like, and traditionally when we are consuming product, I have shown that in soybean, it changes from season to season. So this is a example of a cheese product, again in Himalayan region, which is called chulpi. So when you see the diversity of microbes in different season, both lactic acid bacteria and yeast, it changes a lot. So that is why if the diversity of microbes in traditional fermented food product, it changes, so the effect of those microbes on functionality also it changes. So today what you are consuming, after 2 months you may have similar tastes, but you may have different functionality, and you are not sure that which particular season you are getting a proper functionality. So when you select a starter culture, you should definitely screen and select microbes from different season, see their impact on how it is having impact on your particular metabolite, like anything, either vitamin or peptide or isoflavone or polyphenol, anything. So that actually is tedious screening process should be followed. Now this particular figure, if you see, like the Venn diagram, it shows the earlier one was a microbial diversity. The last slide was there. The Venn diagram, it shows the different season how it impacts the proteomics data. So bioactive peptide, you see AMJ, O and D, October, November, December, January, February, March. It is like different months actually. So we pulled data from different months. We did metagenomic analysis, we did the metabolomic analysis, proteomics analysis, and in different season you get so many different unique peptides, like 373 peptides, 355 peptides. So the uniqueness of these peptides, even though you're getting a similar taste when you consume sample from July or December, but the presence of unique peptides, unique microbes, and functionality associated with microbe, not only proactive peptide but other peptide also, it changes. The second, again, the this one, the metabolomics figure also shows the different season, the metabolite dominance actually, it varies from one season to other. So definitely the health benefit or anything associated with these peptide or metabolite, it changes from season to season. Now, there is one thing called season to season. Now, you can see this data as well. When we collected samples from Ladakh, Sikkim, and Arunachal also, and when we did the proteomics analysis, so the diversity of bioactive peptide, it is so different. But there are some peptides which are common as well. So when you from Ladakh we analyzed actually 1964 peptide, and from Sikkim we analyzed many other peptides. Even though the distance are different, the total peptides we analyzed from both actually is 1964, and bioactive peptide are 108. So among them, these are the 5 peptides we found which is common in both Ladakh and Sikkim. So definitely there are some dominant organisms which are surviving throughout the year, like it can be lactobacillus species, particular species, or some other lactic acid bacteria which are dominant in different season. But there are some organisms which are not present in summer or not present in winter, and that actually impacts the presence of these peptide when they are consumed from one season to another. Now, this particular figure, when you see, we collected samples from different altitude. So the biotransformation of protein in and production of specific bioactive peptide, it differs not only from season, it differs from location, it differs from altitude, because different altitude will promote or impact growth of specific strains. So when these specific strains are produced at altitude of 2000 meter, and some are at 4000 meter, because these samples are collected, if you see the map of Ladakh, there are some samples collected from more than 4000 meters, some are collected from 2000 meter. So we compared, we did the proteomics data of what people are consuming there, and we found that okay, different altitude sample had dominance of different peptide, and there are some peptides. Most peptides are common in many other, but the presence of these peptide, the abundance of these peptide may differ from altitude to altitude, and that again impacts the benefit which you can get from these products like AC inhibitory, antioxidant. So these are different bioactive peptide we identified and compared from different altitude. So what we did actually, we again selected, we screened. I am not showing those data that we screened many multiple organisms, and after screening, we selected 1 or 2 starter culture. And this particular organism, Lactobacillus L4 strain, we applied for production of churpi, both milk churpi and soybean churpi. And this technology was developed. Now we are into scale up of this technology, and again we tried understanding the proteomics of the product which we produced in our lab. So the main challenge with peptide, which would have come to your mind, is so these peptide are prone to gastrointestinal digestion. So if a starter culture is producing peptide which will have this particular property, when this peptide-rich food are consumed, it can get affected by pepsin or chymotrypsin, and hydrolysis of bioactive peptide may be lost actually. But it's not only hydrolysis may lose the activity. Sometimes hydrolysis, there are some peptide, group of peptide, which escapes the gastrointestinal digestion. There are some group of peptide which act as a precursor peptide for production of bacterial peptide after gastrointestinal digestion. So it's not only 1 or 2 things, multiple things which happens during GI digestion. And what peptide is getting absorbed decides the activity of a particular food product. Like when you see this figure, this we did the proteomics analysis for the churpi we developed in our lab. So the one in red, green, and blue. The one in red is the undigested protein, that it is not digested by GI digestion. The one which is green, these bacteripeptides are peptides which are escaping GI digestion. Means they are present in the food. They are also present in the GI digest, like digested GI digested product. So definitely these are escaping the action of pepsin, chymotrypsin, and other enzymes which are associated with GI digestion. And the one in blue are not present in the food product, but they are produced on hydrolysis of peptide which are there in the food product, and these are also bioactive peptide which are produced before absorption. So that's what I was telling. There are different mechanisms which can happen when a peptide in this food product is taken, and actual benefit of that food product depends on what are the peptide getting absorbed on after consumption. So there are the opportunity in bioactive peptide is, like I have shown you some 30-40 bioactive peptide in the last slide. So these are peptides which are already reported. Like when we do the proteomics analysis, I've not shown some specific data, but if you do, when you do the proteomics analysis, you get some 2,000 to 300 peptide in that. And out of 2,000 or 3,000 peptide, only 50, 60 peptides are peptide which are already reported globally in other food products. So there are opportunity for you to explore the activity of more than 95% of the peptides. So the activity of these more than 90 or 95% of peptide is not known. So what we did, we explored the activity of these peptides. We used computational tool to predict the function of the peptides, and after predicting the function of the peptide, we did the interaction of these peptides with target enzyme under which we are seeing the activity, like ACE. We screened them for antihypertensive property. We seen the interaction of these peptide, that how they are affecting or binding to the AC enzyme, and then we synthesize this peptide and check the activity in vitro model, where how these peptide are able to inhibit the activity of AC. Now, this is nothing but a peptide synthesizer, and we check the purity of the peptide which is synthesized. And then these are some of the novel peptide. I have not shown the sequence here because it is some sequence are shown, but after there are some other peptides as well, where which are not reported in other food product, but we have reported in our traditional fermented food product, which are able to reduce the AC activity of AC, which is associated with hypertension. So like this, there are many opportunities of finding novel bacteripeptide in traditional fermented food product, which is produced on biotransformation during fermentation process. So earlier was the peptide which are having single property. Now people are also exploring. This is our product, like fermented soybean hydrolysis which we found, which is also rich in multifunctional peptide. Like peptide, the same peptide will have an ability to inhibit ACE as well as DPP4. So you'll be aware that in population, like when a person has diabetes, there are also possibility he will have hypertension. So such food product which are having multifunctional property, like ability to inhibit AC, which is associated with hypertension, and DPP4, which is associated with diabetes, can be useful for such subjects actually. So these are two peptides which we found which are able to inhibit ACE as well as DPP4. Again, we synthesized and checked their activity in vitro model. Now we are going for animal model to validate these functions. Now, similar to soybean or milk, you can also explore these organisms for a non-traditional fermented food product. Like, right, you don't have a fermented food product from rice bean, but because we isolated these organisms from soybean, we explored how it can be useful for other substrates which are rich in protein. And here again, we developed a process where it can be useful for production of bioactive peptide, which can reduce hypertension. And again, the whole setup of experiment was done to identify these bioactive peptides, study the peptidome, predicting the function, and finding novel peptides. Similarly, we also explored these organisms for production of bioactive peptide from soybean meal, which is again rich in protein and a byproduct of soybean oil industry. And here again, we found some peptides which are able to inhibit AC. So, and antioxidants. So, these actually fermentation process with innovative biotransformation and production of novel molecules with specific health benefit can be useful for value to some of the low value protein rich product. So these are some examples of that. So apart from peptide, there are other molecules which can be produced or which can be improved on fermentation, and the second fermentation which happens in the gut. So see, peptide, the challenge was, or the opportunity was, that this can be getting hydrolyzed by the enzymes, which are digestive enzymes which are produced. But other molecules and like polyphenols or some other secondary metabolites, they can get transformed by the gut microflora as well. So there are double fermentation. One fermentation which you are doing outside, where you are producing cheese or you are producing any fermented food product, and you just imagine the metabolites. You have seen that how vibrant production of the metabolites during food fermentation. So these transformed metabolites during food fermentation, when it is consumed, and they get converted to some other metabolites during gut fermentation. So there are secondary biotransformation which happens in the gut, and that can lead to again improving the health benefit. And that actually has given idea of precision nutrition, where the gut microbes, specific gut microbes in specific community or specific population, can have different impact. Like a food which is produced outside, that particular food can have different impact in one particular population and have different impact on other population. It depends on what is the specific microflora which is enriched in the gut and how it impacts the biotransformative metabolites which is consumed by a subject. Now, this is one example which I wanted to give. Now, if a fermented soybean is consumed, like there is a molecule called digoxin, which is a glycoside actually. It has a carbohydrate link. So the viability associated when it is consumed is less. Now, when it is fermented with bacillus or rhizopus, any organism, it is the glycone form is hydrolyzed to a glycone form. So once a glycone form is converted to a glycone form, the viability with that is improved. Now, this A glycan form, when a fermented product with this A glycan form is consumed, now it is found that 50% of the population is able to convert the A glycan form into equal, like digoxin to equal, and many of them are not able to convert it. So that's why now ones who are able to convert gets equal, which is more active, having more half-life, and having more beneficial. But that benefit you don't get in other set of population. Now, what is the opportunity here? The opportunity is if you can find those organisms from gut which are able to transform digoxin to equal and add that as a co-starter during food fermentation, you can provide equal to everybody. So this is just an example. Such transformation or such things can be looked into different aspect or different metabolite transformation as well. Now, this is one particular example when we produced Bekang and Kinema, and we did the metabolomics, and we found, okay, different strain are having different impact on improvement of specific metabolites. So if you see specific, like this KN1G, KN2G, KN2M, 2B, they have different impact on conversion of digoxin to digoxin. Means converter of glycone form to a glycone form. So it has to start from the beginning. You have to select a strain which does the maximum biotransformation of glycone to a glycone, so that a glycone can be converted to equal. So you can imagine how these microbes at strain level are important to improve your functionality when it is a particular metabolite is absorbed. Now, this is example like when we did the effect of these traditional fermented food in human subject, and we did the microbiome and metabolome analysis of salivary and fecal microbiome. We found that, okay, when these fermented food are consumed, and first what they do, they enrich specific microflora in the gut. So people who consume this fermented food, like I have given picture of fermented food, like most of them are acidic except one which is alkaline with bacillus subtilis. So you see in green part there are some microflora which improves in the salivary microbiome as well, and there are some microflora which increases in the fecal microbiome as well, and there are some microflora which get reduced also on fermented food consumption, and many of them are harmful as well. So there is one challenge, like why controlled fermentation is important. Like traditional fermented food will enrich the beneficial microflora which is associated with traditional food production, but it will also enrich the microflora which you don't want. Like we found improvement of lactobacillus species, but we also found improvement of enterococcus species which is not needed. So that is why controlled fermentation with specific organism is very much important, so that you can avoid such enrichment which is coming from traditional fermented food consumption. So the second part, when we did the metabolomics analysis of salivary microbiome, these are examples of more than 200 subjects actually when we did the study. So if you see the metabolites changes in fermented food consumers and fermented non-fermented food consumers, in the gut we found more than 25 metabolites which are significantly changing, and that again have impact on the health benefit associated with them. Surprisingly, when globally people have not reported changes in salivary microbiome on fermented food consumption because that study has not been conducted. But more than gut, we also found transformation of salivary microbiome. Metabolites, changes in the metabolites in the salivary in saliva actually. So that again, we don't know what is the role it may have, because now these studies has to be linked with some biomedical part or that to come to a real conclusion. But what I want to tell you is, when you consume food with fermented food or non-fermented food, that can have impact on microbiome and metabolite not only in the gut but also in the saliva, which can have different impact on health. Now, similarly, these are the different products which we produced in our lab using different microbes on alkaline or acidic fermentation. And when you see globally, there are many products of starter culture available. And India, we have lot of opportunity because of the diversity which we have. And if we are able to convert some of the traditional food to a global market with proper research, from selection of starter culture to clinical trial proving these products, then it will be really good for the community as well. So these are some of the products with bioactive which are commercialized. Like I told you, product in Calcis in Japan, which can reduce blood sugar. Like similarly, there can be product with for bone health, mineral absorption, or some other cardiovascular problems. Now, apart from this, I just brief about some of the other fermentation or biotransformation which takes place in food and feed. Many of you will not be aware that you get a lot of edible insects as well. Like in different, not only in India, northeastern part of India, if you go, many of the insects are edible insects, and they can have important metabolites which can have important health benefit. Now, fermentation or biotransformation of these have also been useful for production of bioactive or lipids or many other molecules which can have anti-tumor, antiviral, or immunomodulatory effect. Like if you see these are the products which has been even commercialized in other countries, like insect lipids rich in omega-3, like it can be in cosmetics or some other poultry feed, or even for human consumption also it is allowed in some countries. It depends on the regulation of that specific country. Like insect meal is there, which is rich in protein or essential amino acid, which is again many products are available. So people have used fermentation, enzymatic hydrolysis for converting this edible insect into value-added product like bioactive peptide and this insect meal or some other products as well. So apart from food, people have also applied microbes isolated from this traditional fermented food or other ecological niches for biotransformation of this low value agro waste to a high value product. So if you see in India and stands second actually, and the huge amount of agro waste which is produced either from fruit, vegetable, agriculture residues, many, and sometimes it create problem also, like pollution problem associated with them. But if you see, they are very much rich in nutrients, like they are rich in pectin, they are rich in protein, like many of the byproduct, like fish industry byproduct or tannery industry byproduct, or they are rich in many other polyphenols, which can have impact and health benefit if they are purified or if they are recovered from these waste and applied for nutrition, animal feed or functional feed, which is having a huge demand in the current scenario. Now, if like people have applied straticulture or a specific enzyme producing grass microbes for converting this agro waste by solid state fermentation or many other fermentative approach to recover polyphenol, dietary fibers, oligosaccharides, or even bioactive peptide to product having these health benefit. So mostly focus is on animal feed like poultry feed or dog feed, which are actually expensive. If you go to a particular market and if you see the dog feed, most of the dog feed is imported actually, and their price is much higher than normal food also. Now, I'll give you some examples how people have done. Like, for example, this many of you consume fish, and fish industry byproduct. It's create a lot of pollution problem associated with it. But if you consider the amount of fish production in India, the amount of fish waste generation in India, a huge amount of lipid, which is really beneficial lipid, rich in PUFA, omega-3. Sometimes, if you go to marine fishes, it may be rich in DHA and EPA, which is important from brain. So, not you can say that this waste is discarded, but you should also imagine that this much amount of EPA and DHA is discarded. This amount of essential amino acid is discarded. So people have applied fermentation approach to recover lipids and protein hydrolysis from these waste and applied them from animal feed. So this is one particular example. So this is nothing but a tannery waste. Like when you go to a leather industry, the flesh associated with the leather is the muscle actually. That is rich in protein and other molecule, which again has by fermentation and by enzymatic hydrolysis or some acidic hydrolysis, it has been transformed into products which are rich in bioactive peptide and essential amino acid. This is again for animal feed. Like earlier, I've shown how it can be converted into functional food. Now, these are some aspects where agri-waste residues or some other low-cost product can be converted into functional animal feed. Now, this is an example of whey, which is a byproduct of cheese industry, where how it can be converted into products which are rich in bioactive peptides. So, this is again from our research where we identified some novel bioactive peptide, predicted their function and validated their function for antioxidant and ACE inhibitory property. So, this even it is important muscle building and other nutraceutical functional food applications. So India, I told you earlier, we are an agriculture country where a lot of huge amount of biomass is produced. So there is also opportunity of converting this biomass into animal feed or some other, not only feed but other aspect as well. So I have given an example how it can be converted to feed. Like this is where we isolated many microbes from high altitude region and for xylene and cellulose production, and applied xylene recovered from these biomass for conversion of xylene into xylo-oligosaccharide, which is having prebiotic property and also antioxidant property. So these can be converted for functional animal feed with for their value addition instead of burning it and creating pollution associated problems. So these are some data where we are able to produce this biomass from rice straw, corn cob, sugarcane bagasse into xylo-oligosaccharide. We did the scale up of this till 5 liter. We are now going to for higher scale up for transfer of technology for production of prebiotic oligosaccharides. Now, see, microbes are associated with so many. Like I just for your awareness that how yeast what is the source of yeast actually in many traditional alcoholic beverages. So these are different starter culture which is there in northeastern part of India, and they are being preserved. Many of you are microbiologists, but I must say that we generally we complain that okay, we are getting contamination when we do this, we get contaminated. Now there are many traditional people, tribal people in the northern part of India. They are preserving this culture since many generations without much contamination. Definitely contamination is there, but at least they are preserving the key starter, which is doing the fermentation process. Every batch they are doing. So this is nothing but the traditional starter culture which is there, and you can what is our role as a scientist, as a researcher, is that to characterize this starter culture and apply them in industrial process for helping the biomanufacturing of alcohol in a better way, which can help these people to avoid contamination and even export some of the product to the international market. So this is what we expect actually in the biotransformation of fermented food industry. The presently these pictures of are there where we generally go to people and see how it is done actually. Now, if you can see behind us, there is a rack of jute and all wooden racks. So there actually they are doing the solid state fermentation, and below if you see they are burning it for the cooking and all, and that particular is giving the heat, and we generally keep an incubator, but this heat is provided at a higher temperature where this bacillus does the fermentation of soybean. So traditionally, how it is done and what we need in the future, actually, if we are doing more science, understanding the microbes, understanding what the microbe done, selecting a proper strain which does the job, we can definitely go for establishing better platform for production of this food, and which can have better outcome in the market for the local population. So with this, I would like to conclude that what is important and what can be done in traditional biotransformation. See, first to understand what is the microbial diversity, what is the microbial diversity associated with this traditional food, and once you understand that, you select a microbe which does the fermentation, you select a microbe which does improve the functionality. So it all depends on what you need, which metabolite you want to improve, which peptide you want to improve. Specifically, in peptide also, what property of peptide you want to improve. So that if you're doing that, seeing, understanding the mechanism, how it does that, improve the bioactivity and characterize the molecule, see the effect on microbes, gut microbes, and how it is associated with other metabolic disorders. A lot of study has to be done. It's not a period of 2 or 3 years. Huge amount of time has to be provided, and to understand, to bring or to translate this traditional fermented food as functional food for the future. So I would like to acknowledge these people where we visit, and they are so kind enough to make us understand what is the how they prepare traditionally. So that helps us to explore their knowledge, traditional knowledge, for our scientific studies. So once they explain, that helps us to plan our experiment, that how they do it, what is the drawback and what is the benefit. We also spend 1 or 2 days with them, and it is always a kind hospitality which we get. The food which we get, really grateful to them. I'm grateful to the funding agencies which have helped us to do a lot of research in this particular aspect. Either ICMR, most importantly DBT, because I have worked in DBT institutes, and my both institutes, the Royal Society also funded our research. My students, their hard work which I'm presenting here, and collaborators definitely, Professor Andrew Hemmings, where we do the artificial gut facility and other aspects. So if you have any query, not only now, in future as well, you can email me or on similar research platform. So thank you so much. So with more, I hope I was able to provide some glimpse of what we do and what can be done in biotransformation of food and feed industry, and how it can be important for transforming lives of the people who are doing it traditionally. Thank you so much.
Yes, sir. Thank you very much, Amit sir, for sparing the time, and you have delivered wonderful lecture, and you have put several examples of traditional conversion process, and you have well cited some examples of lactic cultures. So now students or GAN participants are invited to ask questions if any. Yes.
So I'm happy I am able to see some participants.
Yes, sir.
Otherwise, online talk we are not able to.
Yes, sir. We are trying to show you some pictures or through the video we are trying. Yes, so yes, please.
Hello, sir. Good afternoon.
Good afternoon.
Sir, you have already discussed in your
It's not
previous.
clear actually. Your mic is.
Hello, sir. I am audible.
Yeah, I mean, it's not so clear. But you can ask. Let me see. I can understand your question. Not. You can ask question.
Okay, sir. Sir, as you have discussed about anti-nutrients in your previous slides, so my question is, how
No, I'm
does
not able to.
Yes, come Anusha this side. Just come in front. Yes. Come this side.
hello, hello, sir? Am I audible?
Yeah. Yes.
Sir, as you have discussed about anti-nutrients in your previous slides, so my question is, how does biotransformation take initiatives in reducing anti-nutrients in cereals or legumes?
Okay. So it all depends that which anti-nutritional factor you want to target. Like if a anti-nutritional factor is proteinaceous, so then generally we apply protease producing organism. One thing I missed actually in my talk, we also work on reducing lactose. Like many people are lactose intolerant, so we apply beta galactosidase producing meat strains for hydrolyzing lactose. So like this, you with the anti-nutritional factor, if you have identified that okay, this particular anti-nutritional factor is there, so you target those anti-nutritional factor using specific biocatalyst producing organism. So that you have to add in your starter culture. So traditionally also it is happening. Like people are keeping it overnight or keeping it for fermentation to reduce the antigen factor. They don't know the science behind it. If and it can be done more efficiently if you select a group of organisms which is doing the job and apply them in a controlled process.
Okay, sir. Sir, is there any project ongoing in your lab or in your supervision that in that particular area to reduce um anti-nutrients in?
Okay, we are working on fermented soybean product where we are working on different soybean seed variety. There are some varieties which is claimed to have not have those antigen factor as well. So we are doing proteomics analysis to understand that how starter culture can have impact on anti-nutrients which are present in soybean. So that one, it's not complete project on that, but one of the objective of that project where we are seeing the effect of that on antigen factors.
Okay, sir. Thank you, sir.
I think, sir, you can stop the sharing of screens. So there are multiple screens are we are seeing this
Okay.
time. So yes, sir. Now it is perfectly fine. So any other question, Jyoti? No. Okay, sir. One more question is यहाँ आइए.
Very good morning, sir. So you discussed about GABA. So
Yeah.
have you done any studies related to check the effect of GABA on cognitive health or in perspective to that?
No, we have not done that. We have only uh, actually GABA. We have only seen starter culture which produces GABA, but beyond that we have not gone actually because our main focus was on bioactive peptide. But definitely it is a people are exploring actually this in animal studies and even clinical trial as well, but we have not done that.
Okay, sir. One more thing, sir. Is there any positive impact of mm, the connection between the microorganism used and the GABA production? Like any specific species that increases more GABA
Yes.
production in some
Yes,
cases?
definitely. Means there are specific species or organism strains which are selected based on GABA. Like we screen for production of GABA, and then organisms which are producing GABA that can be applied for production of product which is having more GABA. So it depends on the specific strain.
Okay, sir. So, one more thing, sir. Which starter culture has been found to produce the most effective anti-hypertensive bioactive peptide during soybean fermentation?
A lot actually. Means people see one thing is there like in India we have this different traditional fermented food product. In Japan they have their own product. In European countries they have their own product. So if we are improving a functionality of a product, it has to be a product which is popular in our population. So like in Japan they have given two peptides, BPP, IPP, and the organism is Lactobacillus helveticus and Saccharomyces cerevisiae. So it's a two culture fermented product. And this VPP and IPP has shown to reduce hypertension even in clinical trial. So that in clinical trial we have not done. So it is difficult to compare with once our studies is also done, then we can say which is better or and that also will differ from population to population. I told you in my talk.
Yes.
So I'm not sure if we'll be able to compare it. We can definitely compare from our product to our product only. We cannot compare with them.
Yes, sir,
Yeah.
definitely. Also, population varies and their gut
Yeah.
microbiota also varies. So, their byproducts also vary. So, it varies from location to location and product to product. Yes, sir. Thank you so much, sir.
Yes, if there is no further query, then we, yes, one more, sir, one more, please. Thank you. Please come forward.
Good afternoon, sir.
Good afternoon.
Sir, as you said that your research on soybean fermented food, so I have a question that what role do fermented soybean food plays in personalized nutrition and gut microbiome research?
Okay, so definitely it has impact on gut microbiome because we have seen also that through our study that people who are consuming this food, microbes which are associated during in this food, they get enriched in the gut. And once these microbes are getting enriched in the gut, the metabolite are getting enriched or biotransformed, it will have different impact on health. But we have in India, we have not reached at that level where we can say what is the impact, which health it is impacting. That still need to be done. Actually, we are much behind than many countries in this fermented food and like European countries, they have done a lot of clinical trials. We still have to do it. We have one or two examples only. So that health aspect part, we still need to work.
Okay, sir. Thank you, sir.
Good afternoon, sir.
Good afternoon.
So, what are the challenges did you face while identifying bioactive peptides produced during fermentation?
Challenges we have not faced much, but one challenges which we are looking for in future is how to trace the viability of these peptides. That when it reaches brain or it reaches, so that definitely is a challenge. That how because we don't know which particular peptide will get absorbed. So if there are peptides which are produced in the body after hydrolysis by trypsin chymotrypsin, it will be difficult to track them. So tracking of these peptides is a challenge. We can see the final bioactivity, but tracking them till the end point is a real challenge.
Okay, so like when we talk about fermentation, it involves like microbial involvement. So, is there any biotransformation technique where we cannot like use microbes instead, any other techniques rather than fermentation?
People also use enzymes
Okay.
for producing bioactive or producing oligosaccharide. Purified enzyme they use for hydrolysis. But I always feel fermentation has a better impact because it will be economical. Like enzymes are a little expensive when you directly apply starter culture. But yeah, again, like fermentation, you cannot control other factors. Like if there is a production of bioactive, there is production of other metabolites as well.
Exactly.
Because the culture produces everything. And in enzyme, it can be precise. If it's a hydrolysis of protein, it is only hydrolysis of protein, nothing else.
Okay, sir. Thank you.
Thank you very much, sir. Now I invite Akanksha to just conclude it, and sir will again resume this session around 2.
Okay.
And Akanksha, over to you. Thank you, sir. Thank you.
Thank you so much, sir, for such an enriching and thought-provoking session. Your talk gave us a deeper understanding of how microbial biotransformation can add tremendous value to our food and feed systems. The insight you shared on microbial process, functional food, and nutraceutical development were truly fascinating and inspiring for all of us. With that, we now come to the end of our morning sessions. I would like to invite all the participants to proceed for lunch. Please enjoy your meal and take a short break. We'll reassemble after lunch for our next session. Thank you.